Okay, and then we were able to see early certain genes, and I will take my word for it, that were important for certain excitatory neurons this is tissue from frontal lobe, which I showed you, is very early affected in the disease. So we're able to look at various regions of brain with this and see what's happening globally with various mRNA, various protein expression. So that's one approach people have taken. Now, they're also looking, and we're not doing this genetically, all the tissues and also are sent to a central bank that is part of the National Institute of Aging Alzheimer program. And they're doing genetic analyses on every one of these, and they're doing analyses of biomarkers. Now, some of these that we maybe found are biomarkers for the disease. They may be selectively increased or selectively decreased. And there's a whole host of these, but I'm just giving you sort of a flavor of where the field is heading. The biomarkers include the A-beta and the tau in its modified form. Those are the best biomarkers, and people look in the spinal fluid, for example, of patients, and they can see changes in the amounts of these. So we're trying to grasp at a lot of genetic markers that may only reflect modest changes in the DNA, only what we call a single point mutation. They aren't big genes that you can assign one gene, one disease. These are subtle. And so these agencies are trying to put this together. And it's very meaningful statistically because every center contributes tissues and serum and CSF to this. So that's a big start in trying to see and distinguish what's happening at that level. Now, we're trying something else too. We send all these samples in, and we've been a big donor of these from, we've had about, must be 700 patients in a certain period of time since they started this program. And we've been able to contribute to that, and all the other centers do as well. So people are really trying to look at the best markers that they can to detect. It's really a fishing expedition in that sense, but this is where it is. Now there's another thing. To detect early, we at USC were very fortunate to have a group from UCLA come over to USC and establish big neuroimaging laboratory there. And I guess they stole them from UCLA, whoever did it. But I've been working with one of the junior faculty people who does this really elegant, high-resolution neuroimaging. And they were able to map out certain changes using their markers. And I'm not an expert in neuroimaging, but that's why I have the other person, and we're working in a partnership on that. And we just recently got a wonderful grant to start proving something I've been hypothesizing as well, and I'll get to that. One of the important things that happens if the synapse, the end terminal with touching the next terminal, dies, is you lose neuronal connectivity. So one neuron, like in the hippocampus, does not touch another neuron anymore, and it short circuits it. So perhaps, and that is hastened by the abnormal tau protein in it, or the A-beta, which is toxic. We're not sure really what's causing it in toto. But there's much more detail that I'm not telling you about that. I just want you to get the general idea. So this recently, about a year and a half ago, a group at Stanford who do what's called optogenetics, that means catalyzing the role of light in different light power and wavelength, catalyzing reactions. They've been able to define neuronal connectivity in the following way. The group is a lab at Stanford. The head of the lab is this fellow, Dieseroth, And his publication, it wasn't on Alzheimer's, RI, and we're now able to do this technique that is allowing mapping of neuronal connectivity in a tissue volume of human brain. Now, we look at these histologically normally to do our diagnosis, and the histology is done on like small, thin pieces, 5 microns, so you can see one neuron how it's touching the other. But this allows you to look at maybe, and in fixed tissue, not just frozen, a piece 5 or 6 millimeters. So we go from millimeter down to micron size. And so we would be able to see a larger area and how that connectivity using immunohistochemistry against the markers that I've shown you, against the tau, and you saw some of that, with the tangles, and against the A beta, and against other markers specific to the neurons that are involved in the disease. And that's immunohistochemistry. They detect proteins. You can look at the mRNA. So some of the ones that we came up with is modified in the disease with our panel that I showed you, with what we call the heat map. The mRNA can be looked at by what's called in situ hybridization. So we can localize that in the tissue. The problem is, so this is big, and it might span the distance between a small neuron, a big neuron that's involved, to the next order, and the next in the chain of neurons that are involved in the disease. So the problem is how we fit this together. You can't take little slices and then look at it by imaging, which is at the millimeter level, because there's it doesn't fit together. They're so small you wouldn't be able to, the axons or the processes, you wouldn't be able to follow one and see where it goes into the next section. But if we use this type of mapping in a big tissue, thick tissue, we should be able to co-register these and compare the histology in the volume with by co-registration with the imaging. And I'll show you there's very fancy imaging that they're able to do, and we're now doing. And we take a piece of tissue. This is very novel. We've just gotten funded to do this. Here's the hippocampus right here. It looks like a seahorse, just like it's named. I put little glass markers. These are just tubes for hemocytometry. Put them into here. This is just here's one right here and one right here. And here's the hippocampus. And that's, we then do the high resolution, what's called diffusion tensor imaging. That works by water flux through the processes that connect each other. And then we get those on the same piece. Now, people haven't really been looking at autopsy tissue, and we have here. And we're able to see really great stuff on it. And that's why I think we were getting our money for it. And this then is put through the Dieseroth method, and we can then look at it. It's clarified, and that's an interesting thing, but I won't get to that. Here's the hippocampus, and here's the area near it. This is done with 2-photon microscopy, and we're looking at this little piece right here. And here we see stained with the antibody against the tau, the neurofibrillary tangle material. We see it right here. That's abnormal. That's the Alzheimer. Here's a cell where it's plugging up the cell. So we're going from gross here, the big one, and we can see it here, and we have other ways of looking at it that my colleague does. And here we can see it at the micron level. So we'd be able to look in here and correlate what's in here. So I think that's going to be cool when we put it together and see this, how it's going to actually, you don't have to read all of this in here. Don't try. But I just want you to get the idea. We can take the same piece and merge it together. The idea ultimately would not just stick with this, which is post-mortem, but to look at it in vivo on patients. That's our goal on this. So we have to define this. If you can see this in this earliest place where the disease will occur, then in vivo we should be able to see how far that patient is. And with our goal would be with agents for treatment that you could see that change. So that's one of the so-called cutting edges that we're working on here. And the person who does the neuroimaging can actually look at, this is the same piece I showed you. And here we can see the regions of the hippocampus. We know the impulses come into here, they go here, to here, to here, here. And here's with the neurons, and this is mapped out, and they can do, look at how this is a connectivity map of this region here is this region here, which is over in this region here on that cartoon. So we should be able to see how this changes, how the connectivity changes in the earliest part where the disease starts. And that is our goal on that. And here, this is a T2 MRI imaging on the same piece. And we should be able to, and here's the co-registration. We can see where the tubes were. That's just a little hemocytometry tube, sheet, and it can go through all the processes. It's not distorted by the magnet in imaging. It's not distorted when we put it through for clarity. We tested all that. And so this will be very nice if we can, we're just at the beginning. So you can't see results of this yet, but this will help us in vivo, hopefully, see where this patient is clinically. And that's a start in getting an early diagnosis to see if it fits. If it's another disease that causes dementia, and there's frontotemporal lobar dementia, which is quite different, we've been able to separate these early. So that's what our goal is in that. This is how we do the co-registration. These were the holes, and she's putting it, she has a planar view of the MRI data. It's superimposed here where they overlap at about 300 microns. It's superimposed on the tissue, the tissue that we saw. Now let me go on here. And this is another view of how these become superimposed. This is the tissue, 2-photon microscopy. And it just superimposed the same piece, and here we know they're in the same place. So that's just showing you one of our approaches. It's not the only. I could tell you many, many, many more things that are going on in the field, but I mean, nothing is, it's going to have to be sort of put together, and nothing is entirely clear yet. But you want to get the diagnosis early. You have to know exactly which molecules are involved. We know what to look for. And with treatment, we should be able to clarify what this is over time or with different stages of the disease.